Hello everyone, my name is Sonali. Welcome to the channel Success Edutech. Today we are going to see the explanation of the concept of dominance from the lesson Inheritance and Variations of 12th Biology. Now, please watch the complete video. Please like, share, subscribe to our channel. Link is given in the description. Please watch the previous videos to increase your knowledge of this lesson. Every video is in detail, so please watch the complete video. Now, we will see what is the explanation of the dominance, concept of dominance is that every gene contains the information to express a particular trait. In previous videos also I explained this that every gene has consisting of particular trait to transfer that trait towards the next generation. In a diploid organism, there are two copies of each gene. This you know, two copies of each gene, that is known as a pair of alleles. Now, these two alleles need not always be identical. Example, they will be heterozygote. Now, let's take an example. Suppose a gene that contains the information for producing an enzyme. And now there are two copies of this gene which can produce an enzyme, that is known as two allelic form. Let us assume, as in more common, that normal allele produces a normal enzyme that is needed for the transformation of a substrate S. Means whatever there will be the enzyme formation. Now, theoretically, the modified allele could be responsible, the normal allele and the modified allele. So, modified allele could be responsible for the production of first normal and less efficient enzyme or non-functional enzyme, then no enzyme at all. So, it can be responsible for that. In the first case, the modified allele is equivalent to the unmodified allele. It is the equivalent. That is, it will produce the same phenotype trait. Equivalent means it can produce the same phenotype same character, that is result in the transformation of substrate S. Same character, whatever allele is produced and modified allele is also produced and the normal allele. Such equivalent allele pairs are very common. But if the allele produces a less functional enzyme or a non-functional enzyme or no enzyme, the phenotype may be affected. But if allele produces less functional enzyme or non-functional enzyme or no enzyme, meaning is the same, the phenotype may be affected. So the phenotypic trait will only be the dependent on the functioning of the unmodified allele. Now, modified and unmodified allele means what? The unmodified allele, which represent the original phenotype, is dominant allele, which represented or gives us a dominant allele, and the modified allele is generally the recessive allele. So that is the difference between the modified and unmodified. Unmodified means it can produce a dominant allele, and modified it can produce a normal allele. Now in this video I am going to explain you one certain basic factor, that is a polygenic or quantitative inheritance. Means what? What happens to this in human population? Characters such as height, skin, color and intelligence show gradation, means continuous variation in expression, and not only two contrasting characters. These characters are determined by two or more gene pair. So that gene pair, they have additive or cumulative effect. Means the genes are known as cumulative genes or polygenes. It can be known as polygenes or multiple factors also. Means the ratio it can come into 1:4:6:4:1 if there are two genes having the additive effect. Example, wheat kernel color. And in case of three such genes, multiple allele, polygenes, the ratio it will be 1 is to 6, is to 15, is to 20, is to 15, is to 6, is to 1, whatever it may be ratio. Example, skin color in human. So this wheat kernel color, Swedish genetics and the Nissan in 1920 discovered the polygenic inheritance. He crossed red kernel variety of wheat with a white kernel variety, and in F1 generation all plants had grains with the intermediate color between red and white. That is, and in F2 generation, what happened? 5 different phenotypic expression appeared in the ratio 1 is to 4, is to 6, is to 4, is to 1. So it can be red, intermediate and white in the ratio 1 is to 2 is to 1. Continuous variation in the expression were as follows. The darkest red as red as the parent plant, suppose 1 by 16, then medium red also, then intermediate red also, light red also, white also. So this Nelson L suggested that the kernel color in wheat is controlled by two pairs of genes, that is capital A and small a and capital B and small b. So it is a phenotypic ratio, it would be 1 is to 4 is to 6 is to 4 is to 1. So it will be the graphic representation of cross between red and white wheat kernels showing polygenic inheritance. So in this video I am going to explain these, that gene A and B determine the red color of the kernel, and capital B and dominant over the recessive allele small a and b, which do not produce a red color pigment, and the expression is white if dominant gene is not present. The genotype of parent with red kernel is capital A, B, T, and that of parent with white kernel is small a, B, B. So after crossing, what we will get? The genotype in F1 hybrid is capital A, small a, capital B, small b, and during gamete formation, each gene pair shows a Mendelian segregation and 4 types of the gametes are produced. So continuous variation in expression is observed as each dominant gene is produces specific amount of pigment and the shade of red color. Depends on the number of dominant gene present as shown in the Punnett square. So only 1 out of the 16 is of the 4 dominant gene, and this is the darkest red. So 4 out of 16 have 3 dominant gene, and our medium red 6 have 2 dominant gene, and our intermediate red 4 have the intermediate red and 4 have only 1 dominant gene, and our light red and again only 1 is without any dominant gene and is therefore with the white kernel color. So that is the example of the red white kernel. Now, which are not always be identical, we will see the MCQ related to this topic. 2 genes, 2 alleles, then 2 character, then none of these. And the answer is, now these 2 alleles need not always be identical. Example, heterozygote. And the answer is 2 alleles. Now second one is, if the allele produces a less functional enzyme, so phenotype may be affected, genotype may be affected, phenotype may not be affected, genotype may not be affected, and answer will be phenotype may be affected. Now, thank you very much students for watching my complete video.